it's Delaney James. I know my account still says not in. However, James is my new last name since getting married. In this presentation, we are going to cover electrical pacemaker activity on autorhythmic cells in our heart. This is for Grand Canyon University Bio 520 class. So, what are the objectives of this presentation? As always, it's good to know where you are going and what you should hopefully learn out of this presentation. The first thing to understand is what exactly are cardio autorhythmic cells, what is pacemaker activity, and then how do funny channels, potassium channels, and calcium channels play a role in pacemaker potential. And then we're going to look at a research article done that helps to support the calcium clock as a main role in controlling rhythmic contraction of the heart. So, first off, what are cardio autorhythmic cells? The heart is a major organ within the body, and it is obviously responsible for pumping blood to all of our organs and our extremities. The heart must pump rhythmically via an action potential that is produced by itself, and that property is called automaticity, meaning it is automatic. There are two specialized cells within the heart that include contractile cells and autorhythmic cells. Contractile cells make up 99% of the cardiac muscle and achieve the actual pumping of the heart. Autorhythmic cells, on the other hand, make up the rest of the heart and they do not contract. However, they do initiate and conduct the action potential for the contractile cell. So these cells are going to serve as the pacemaker in initiating the cardiac cycle and provide a conduction system to actually synchronize or organize the contraction of muscle cells within the heart. So, what is pacemaker activity? Cardiac autorhythmic cells don't have a resting potential, meaning they don't have a non-excited state due to the electropotential difference. Instead, they display something that is known as pacemaker activity. This means that the membrane potential is going to slowly drift between our action potentials until threshold is reached, and then the action potential is fired, and then this repeated cycle of drift and fire causes our cells to cyclically initiate an action potential. The action potential will spread out to the heart to trigger a rhythmic contraction. And this is different from our other cells because the nervous system does not need to stimulate the contraction. If you look at the graph up top right here, this is what we normally think of when we see an action potential, either in our skeletal muscle or like maybe you're picking up something, your action potential needs to be moved around. All right, we sit at resting right here. We reach the threshold. We are going to become positive and depolarize. We'll repolarize as we become negative. We get a little too negative and hyperpolarization, and then we are right back to resting until the nervous system sends another signal and we can start the cycle over. Down here, however, is what it looks like when we're talking about the heart. Right? We never really sit at our resting. We are going to depolarize, repolarize, and right here we have this drift between hyperpolarization and the next depolarization. This drift right here is what's going to allow the heart to contract over in a cycle rather than having to wait for a signal for the heart to contract. It's going to actually depolarize itself to reach that action potential. So this is an overview of what pacemaker potential actually looks like. Pacemaker potential is what we refer to as the slow drift, that graph on the last page, that slow drift to the threshold that was just mentioned. The first part of pacemaker potential is actually going to be the result of simultaneously opening of the funny channels, which we'll get to in a minute. This is going to allow sodium ions to flow into the cell, so that's number one. Potassium ion excretion is going to decrease, so potassium stops leaving the cell. Those channels are shut. Eventually, calcium ions will flow into the cell, which is going to allow the threshold to be reached. And then we have calcium ions that are going to allow the depolarization and repolarization of the cell to occur with potassium as well. Right, our positive charges leaving and entering the cell in order to make the cell positive and then become negative again and restart the cycle over. We're going to look at each one of these steps kind of in detail. We're going to start with the funny channels because this is very important in cardio autorhythmic cells because they're essentially setting the tone for the whole contraction. The slow depolarization to the threshold, that little ramp like below the threshold right here, is going to be caused by sodium ion entering through a specific type of voltage gated ion channel that is only found in cardiac autorhythmic cells. No other cells have them except for these. All right, normally what's going to happen during a action potential is voltage gated channels will open when the membrane is going to become positive. However, these cells are kind of the opposite. So when these channels are going to open, it is when the potential becomes more negative during hyperpolarization. So this dip down, we get more negative. Our funny channels are going to open. They're going to allow sodium to enter the cell, slowly becoming positive to reach that threshold and send the next action potential. These funny channels are the reason the heart never stops beating. At the end of one action potential, when it hyperpolarizes, normally the membrane potential returns to resting until another stimulus comes along. However, if the heart stayed at resting potential, the heart would clearly not be beating. So when one action potential ends and hyperpolarizes, funny channels open, making the cell membrane potential positive by the influx of sodium, moving the potential towards threshold. There are two other key ions when we're talking about heart contraction, and that is potassium and calcium. So we need to mention them when we are referencing um, autorhythmic activity. In cardiac autorhythmic cells, the permeability to potassium is not going to be constant between the action potentials. The potassium channels that are open during the potassium channels are going to open during the repolarization of the last action potential. So on our way down, we're going to see calcium channels open, which will slowly it will slowly close at negative potentials. This slow closure is going to weaken the outflow of potassium down the ion's concentration gradient, and the slow decline in the potassium outflow occurs at the same time as the slow inward leak of sodium through our funny channels that we just talked about on the last slide. Calcium has an increased entry to the cell, so our funny channels close and transient calcium channels open before the membrane will reach the threshold. This resultant brief influx of positive calcium will further depolarize the membrane, once again bringing it up to threshold. It's referenced here where it says Ca2 plus, that's calcium ion coming in, sodium's coming in due to the funny channels. It's going to bring it to threshold. Once that threshold is reached, the transient calcium channels close. The action potential is going to fire. Calcium is going to come out. That's what I was talking about earlier. Potassium is going to come out until we reach the negative, and then we're back over here with sodium and calcium flooding in. And this is going to occur over and over and over again because remember that heart can never stop beating. If we take a look at something called membrane clock mechanisms, really there's one whole one called the membrane clock mechanism, and then there's another one called the calcium clock. We're going to briefly reference those two, and then the research page on the next one will go ahead and overview the calcium clock. But these ionic changes with potassium and calcium and sodium and the membrane potential are going to cyclically bring autorhythmic cells to the threshold. This is known as the membrane clock mechanism. This mechanism was thought to be responsible for pacemaker potential and the rhythmic beating of the heart all on its own. This idea of sodium coming in, sodium coming in, sodium coming in, calcium coming in, and we're going to do all this until we reach the action potential ignition, right? This is the membrane clock model. The calcium clock mechanism, however, acts with the membrane clock to excessively self depolarize the membrane. Down here, we're going to get that self depolarization. Once the threshold is reached, the depolarization phase of the action potential is going to occur in response to a long lasting voltage gated calcium channel. And this large influx of calcium ions is going to bring the cell membrane potential to being positive, swinging the potential towards a positive direction. And this potential is due to the outflow of potassium and the closure of calcium channels. As discussed earlier, once our membrane potential is over, the slow closure of potassium channels is going to contribute to the beginning of the next action potential. So it's simply going to just kickstart this depolarization in order for our cell to reach the threshold and send an action potential. So is the calcium clock the main driving force, or is it the membrane clock mechanism? So a study by Maltsev and Lacta, I think I butchered that guy's name, um, they looked into the calcium clock and its relation to the heart rhythm of our cardiac cells. And they initially state that pacemaker cells were driven by voltage gated ion channels known as membrane clocks. This study collected experimental data and they mostly used little rabbit cardiac tissue and looked at the nodes within those tissues and how they responded to initial ion concentrations. And they also reviewed simulations multiple times to demonstrate that rhythmic contraction is due to the intracellular calcium clock. Studies, just like this one, show that rhythm is initiated, sustained, and regulated by fluctuating calcium ion releases from our sarcoplasmic reticulum, which is a calcium storehouse within muscle cells. This release of calcium is driven by a high level of basal phosphorylation, or calcium cycling proteins, according to this study. The release produces calcium little wavelets under the cell surface membrane during depolarization, and activated sodium and calcium exchange, and calcium and sodium exchange is activated, until membrane potential ignites an action potential. Cardiac pacemaker mechanism is a very complex one, and it includes a balanced integration of intracellular and membrane delineated processes. The picture that is on the slide was referenced in the paper. It was to the side a little bit. However, I did find it on another source as well, so I'm not sure which one came first. But the source that I found this picture on as well kind of explained it a little better, showing the influx and efflux of calcium and sodium ions that I just explained and how they're going to change the membrane potential to drive towards the action potential. So overall, we see that there is a influx and efflux of sodium, calcium, and potassium um, through voltage gated channels, and more specifically, sodium through the funny channels that are going to allow um, the cardiac autorhythmic cells to drift towards that threshold that they need to reach in order to continuously send the action potential, telling the heart to pump in a rhythm over time. This nervous system is not needed. It is all self sustained due to ion concentrations. Some people believe that it is the calcium clock that is the main driving force. Others still believe that it is the membrane clock mechanism. However, there is no denying the fact that all three of these ions have to be present and play a role with each other in order to allow our heart to pump. These are my sources. I hope that you enjoyed this presentation and you at least learned something. This will be on YouTube, so if you have any questions, leave a comment and have a blessed day.